Hey all, in this video lecture we're going to talk about mutations. Particularly, we're going to talk about what are known as point mutations. But before we get to this, let's define what a mutation is. Mutations are changes in a DNA sequence that are heritable. In other words, these are changes that can be passed down from a mother cell to daughter cells. These changes can be considered to be good, bad, or neutral in that they have no effect. But it's really a matter of perspective. For example, when we talk about antibiotic resistance in bacteria, the mutations that occur that allow for bacteria to grow in the presence of an antibiotic are good for the bacteria. However, for us, we consider these to be bad because, well, as the name suggests, it leads to resistance of these organisms to antibiotics that we use to treat them. Now, again, the types of mutations that we're going to talk about in particular are ones that are called point mutations. These are single base pair changes. And we'll take a look at 3 different examples of point mutations. Mutations can occur by a couple of different routes, but in essence, all of them occur by mistake, essentially. Mistakes that occur during replication are the cause of mutations. And the mistakes that we talk about are ones that are caused by DNA polymerase. DNA polymerase isn't perfect in making a copy of DNA. Sometimes it can make a mistake. Now, there are things that can increase this rate of mistakes that are made by DNA polymerase, and these are called mutagens. Mutagens are any chemical or other sort of source that causes mutations. Two that you may be familiar with are UV radiation and ionizing radiation. Both of these cause issues with DNA that when DNA polymerase goes to replicate the DNA, it often will make a mistake where UV radiation or ionizing radiation has disrupted the DNA sequence. And therefore, both of these can increase the rate of mutation. So, what we're going to take a look at now are 3 different types of point mutations. But in order to identify those mutations, we need to start with a template DNA sequence that's going to represent our original state of DNA. And that is going to be represented in this box here at the top. You'll notice I've listed the template DNA in a 3' to 5' direction. And remember, template DNA is antiparallel and complementary to an mRNA sequence. So what I'm going to have you do at this time is pause the video. First, figure out what the mRNA sequence is that is generated by this template DNA, and then go ahead and figure out what's the amino acid sequence coded by that mRNA. You see here that the mRNA sequence that is generated by this template DNA will be from the 5' to 3' direction, AUG, UUC, AGA, and CGG. And you can notice here what I've done already is I've used these little blue marks as a way to indicate the breaking between the codons of the mRNA. And if we use our codon code wheel as a way to analyze the sequence of mRNA, then we find that the amino acid sequence produced by this mRNA will be MET, Phi, ARG, ARG. Now let's look to see what effect do mutations have then on the mRNA and the amino acid sequence. If you compare our original sequence to the sequence in the bottom left hand here, what you're going to find is that the 6th letter has been changed from a G in the original sequence to an A in this template DNA sequence. If we write out our mRNA sequence, we'll start out with AUG again, and now instead of UUC, we're going to have UUU, AGA, CGG as our last 3 letters. If we put natural breaks again in the mRNA sequence, like so. Our first amino acid is going to be exact same. And now the second one is where we have this new codon. Previously, our codon was UUC. It is now UUU. However, both this and the original codon coded for the amino acid phenylalanine. And our last two amino acids have not changed. And so this is indeed a mutation. It's a change in the DNA sequence. But we categorize this type of mutation as a silent mutation. Silent mutations are ones that change the DNA sequence, but they have no effect on the amino acid sequence or the structure of a protein. At this point, what I'd like you to do is figure out the mRNA and amino acid sequence that's produced by the template DNA in the middle of the three blocks here at the bottom and on the right-hand side. Go ahead and pause your video now so that you have time to do that. All right, so now is the time to go ahead and compare your sequence to the ones that I've written. If you're looking at the middle box here, you'll notice that it's the fifth nucleotide that has changed. Previously, in the original sequence, it was an A, and now it has changed to a T, as shown here. And this then, again, is going to change our second codon. Before, where our second codon read UUC, it now reads UAC. And the effect you can see here is that it's going to change the amino acid. Our original amino acid in the sequence was phenylalanine. We now have tyrosine. And so this is an example of what we call a missense mutation. Missense mutations are ones where the DNA will change, and this change results in a different or new amino acid. All right, so let's take a look at the last of these, which is over on the far right-hand side here. You can see that I've already underlined in black the nucleotide that has changed. Previously, in the original sequence, it was a T, and now it is an A in this new sequence. This has created a new codon. Previously, our third codon was AGA, and now it is UGA. And if you carry this out to look at the amino acid sequence, this is actually coding for a stop codon. And so we call this type of mutation a nonsense mutation. The name comes from the fact that sometimes stop codons are known as nonsense codons. So just like any of these mutations, it's going to change the DNA. And that change in the DNA will create an early stop codon, or one that's earlier than is to be expected in the amino acid sequence. So in summary, what we've looked at here are changes in DNA, and again, these changes from one generation to the next are known as mutations. We've talked specifically about point mutations in which one nucleotide is swapped for another one. There are additional types of mutations, known as insertions and deletions, that also will affect DNA sequence, mRNA sequence, and amino acid sequence. Key takeaway from here is to note the difference between these three types of mutations that we've talked about. Silent mutations have no effect on the amino acid sequence. Missense mutations change an amino acid in the sequence, and nonsense mutations create a stop codon.